This will be our first clinical correlation lecture of the course. Our normal clinical correlation lectures will be a little different than the principal lectures. In the principal lectures, we'll discuss basic fundamental mechanisms of classical and molecular genetics, but in the clinical correlations, our goal will be to examine the way that those principles can deviate and how that deviation leads to various forms of genetic disease. So typically in a clinical correlation, what we will do is we'll take an example disease, we'll look at its presentation, its signs, and its symptoms, and we will look at the way that the underlying genetic process has been altered and how that relates to the development of the disease and the specific signs and symptoms. This one is going to be a little bit different. We're going to go over pedigrees as a tool. We'll remind ourselves about the nomenclature and signs and symbols of a pedigree, and we'll look at the way that pedigrees can be used to analyze genetic transmission. It's been said that humans are both the best and the worst organisms for genetic study. When we think about humans, we have spent more time and money on understanding their physiology, anatomy, etc., more so than any other species. So the underlying structure and function of the organ systems individually and the organism as a whole is clearer in the human than anything else for obvious reasons. The problem, of course, is that there are many questions about genetics we would like to know the answer to that we cannot for ethical and practical reasons. This is why, for example, when we go through the principal lectures, you'll notice a lot of the examples that I'll use are in animals. And you may be tempted to ask, why are we looking at these examples in animals in a medical genetics course? And quite frequently, the reason we are is we are not able to examine some of these principles in humans the way that we examine them in plants and in animals. And so we discover the principles in these other organisms and then relate them to what may be happening in the human. So we're sort of caught in this weird place where we know a lot about human physiology, biochemistry, etc., but we can't do the sorts of studies that would answer the question. One tool that helps us sort of work around this issue is the pedigree. The pedigree allows us to answer some of those questions that we'd like to be able to answer, that are very easy to answer in an animal system that are not able for us to address in a human system. So a pedigree is a pictorial representation of a family tree that zooms in on the inheritance of one or more characteristics, typically a genetic disease that's being done at the request of a family member with a genetic counselor to lay out likelihoods of the transmission of an underlying error. Now, I know that you've used pedigrees in other courses probably multiple times. I just want to remind us of what the nomenclature is. Squares are representing male family members. Circles represent female family members. Affected persons have filled-in shapes. Carriers have partially filled-in shapes. And then there's a couple of other things that are less common. For example, the proband has a P with an arrow with it. That's the person who's come to the geneticist or to the counselor and says, hey, I'm affected, or hey, my husband, my wife, my child is affected. That's the person who's seeking the analysis. When we think about the way that a pedigree is structured, vertical lines denote genetic transmission. Horizontal lines represent members of the same generation. You can have two lines, a double line that represents consanguinity, which is a mating between related persons. So when we're thinking about analyzing pedigrees, one of the things that is challenging is that oftentimes the probabilities that we expect to see are not observed because the number of members of the family are relatively small. So for example, if you think about the probability of flipping a coin and getting heads, that probability, assuming that the coin is a fair coin, is 50%. But if you only flip it once and you get tails, your data says 100% of my data points is tails. Even though it should be 50-50, 100% is tails. And so that illustrates the problem, right? If we're dealing with one child in every generation, it can be difficult sometimes to see the types and the proportions of phenotypes that we expect. If you go back far enough and you have enough data, most of the time it's still pretty easy to figure out what type of transmission you're looking at. But you just need to be aware that it may be slightly off the proportion that you expect it to be. Now, what I would like to do from this point on is look at a couple of different patterns of pedigrees as they relate to transmission, whether we're talking about autosomal, dominant, or recessive disorders, and then sex-linked disorders. So when we think about autosomal dominant disorders, and we're looking at a pedigree, what sort of patterns should we expect to observe? If it's autosomal dominant, that means that it's a gene on one of the non-sex chromosomes, and therefore the sex of the patient should not influence the probability of having the particular phenotype, which is another way of saying we should expect males and females to have the same proportion in theory. Now, once again, going back to my example about the coins, right? Flipping that coin twice and getting two tails does not necessarily mean that the probability is not 50%, but it should appear equally in both sexes. Because it's a dominant disorder, it shouldn't really skip generations. You should be able to find every affected person has an affected parent. And we'll see this in a moment. We're going to talk about some of these disorders in this course. For example, we've got a clinical on both Waardenburg and HHT that will come up in a later lecture. So here is a pedigree showing the pattern of autosomal dominant disorders. So when I'm doing this, I ask two questions. Number one, is the proportion of male to female roughly equal? So we can count 1, 2, 3, 4, 5 affected females, 1, 2, 3, 4, 5, 6 affected males. That's essentially equal. The next question I ask is, does every generation have an affected individual? Or another way of saying it is, does every affected person have an affected parent? Now, we don't know anything about number two's parent because we don't have records that go back that far. But we look in generation 2, this person is affected and they have an affected parent. This person is affected and they have an affected parent. And as we look at each affected person has an affected parent. And that is a pattern we observe in in a dominant disorder. So we answer the question, is it autosomal or sex-linked, by looking at the proportion of male to female. And in this example, it's essentially even. That suggests autosomal. We answer the question of dominant and recessive by saying, does every affected person have an affected parent? Both of those questions point us towards an autosomal dominant pattern of transmission. What about autosomal recessive disorders? Now, once again, because it's autosomal, we would expect them to be equal between males and females. An affected person, because it's recessive, must have received a recessive allele from mom and a recessive allele from dad. That's how you get a recessive disorder. If the trait is rare in the human population as a whole, it may skip generations. And this is because you'll have non-affected heterozygous carriers. So if it's rare, then the likelihood that the person you marry outside the family has that rare allele is low, meaning most of the people that marry into the family should be homozygous dominant. You could get a carrier, of course, but the probability is low. And because it's recessive, unaffected individuals can pass on the trait because they're carriers. There are many, many examples of autosomal recessive disorders. Some of them we'll talk about in clinical lectures. For example, we'll discuss Tay-Sachs disease, and sickle cell and cystic fibrosis will come up in our lectures. So here is a diagram illustrating autosomal recessive transmission. So once again, let's ask the two questions. What is the proportion of male to female? In this case, there's one affected female, one affected male. That suggests autosomal, but my N value is only 2, so I have to hold that kind of loosely in my mind. And then I ask, is the trait found in every generation, or does every affected individual have an affected parent? So number 3 is affected, but neither parent has the disorder. Number 2 in generation 4 is affected, but neither parent has the disorder. So if we think this through based on what we know about how Mendelian transmission works, 1 and 2 must both be heterozygous, and they have one child that received from each of them the recessive allele. Now, if 4's grandson has the trait, that means that 4 must be a carrier and then passed that allele on to his daughter, who then passed that allele on to her son. And then the same way here. This individual passed the allele to her son, who passed it to his son. Now you'll notice here that number 3 of generation 3 and number 4 of generation 3 are first cousins. And here's the double bars that indicate a close pairing, a consanguineous mating, and therefore their son is affected. You can have the transmission of recessive disorders without mating within the close family. But mating within the close family increases the likelihood that that happens because the family already has the trait, and it's much more likely that you get it from within the family pool than when someone from the outside marries in. Okay, let's switch gears a little bit and talk about X-linked recessive disorders. Now, because X-linked recessive disorders are found on the X chromosome, males are more likely to be affected. And this is because males having an XY pattern only have one X chromosome. And so if they receive a recessive allele, they cannot be quote unquote saved by another X chromosome with the dominant allele. We say that males are hemizygous, which means they express whatever allele they have on their X chromosome. So you would expect an X-linked recessive disorder to be more frequent in males than in females. There should be no father-to-son transmission, and that's because, of course, the father gives to his son the Y chromosome, not the X chromosome. If the mother is affected, so if the mother is homozygous recessive, all of her sons will have the trait because she only has an X chromosome with the recessive allele, therefore all of the children, all the sons will have it. If the mother is a carrier, we would expect half the sons to be affected and half the sons to not be affected. Many examples of this you're probably aware of. Some of the most famous are hemophilia, muscular dystrophy, and red-green color blindness. This is a chart of the royal families of Europe leading up to the conflict of World War I. What you'll notice is that this is a chart of the transmission of hemophilia. Queen Victoria of England was a carrier for hemophilia, and she passed that trait throughout her children who married into the royal families of Europe. And so when we think about marrying into the royal families of Europe, this is an artificially constrained breeding pool. So in 1910 and 1890 and 1880, if you're a child of Queen Victoria, you can't marry anyone. You can only marry someone within the other royalty of Europe. And so you're constricting your dating pool, which means you are increasing the likelihood that you transmit a recessive allele. So if you look at Queen Victoria, she had a daughter named Alice, who was a carrier, who went on to have an affected son. She had a son, Leopold, who had hemophilia and went on to have a daughter who was a carrier and then that daughter who was a carrier had an affected son. And so you can see the way that this spreads. Now, once again, going back to our two questions. First, what is the proportion male to female? Well, if I look at this chart, the only people who are affected are males. So it's definitely not equal. And because it's all males, that strongly suggests it's X-linked, recessive. Now, once again, if it's recessive, affected people should have unaffected parents. So if we look at Leopold, his parents are unaffected. Leopold and Maurice, same thing. Rupert, same thing. All of the sons have unaffected parents. Now notice it doesn't skip generations. It's present in generation 3, in generation 4, and generation 5. But nevertheless, if we look at the individuals, it skipped this generation, but it's found within the whole generation. So Leopold had it, his daughter did not, the grandson did. So the fact that we have affected people with unaffected parents, the fact that it's all males, that argues very strongly it's an X-linked recessive disorder. Okay, what about X-linked dominant? So this is sort of the reverse. Females have 2 X chromosomes, and therefore they are twice as likely to inherit a dominant allele. So it's almost like if you think there's a deck of cards, right? And in that deck of cards are some cards that say you will have the genetic disorder and some that say you will not have the genetic disorder. Males only have to draw 1 card. They get 1 X chromosome. So the probability that they draw a dominant disorder is less than females who have to draw twice. It will appear both in males and females, of course, but more frequently in females for that reason. Because it's dominant to have the trait, you must have an affected parent. This also means that an affected father will pass it to all of their daughters because he only has that 1 X chromosome to give, but he cannot pass it to his son. Now, there are scenarios like Klinefelters and non-disjunction, and we'll talk about those in other lectures. But these rules are assuming normal chromosome count in the children that are produced. If you have what we call an aneuploidy, which is a person that has the wrong number of chromosomes, instead of 46, they have something else. We're going to ignore that scenario for these rules, and we'll talk about how that impacts transmission in a later lecture. Once again, because it's dominant, it should not skip generations. So here's a chart showing a X-linked dominant pattern. So once again, let's look at our proportion. 1, 2, 3, 4 males. 1, 2, 3, 4, 5 females. Now that's pretty even. We've already said that, we had another example where it was 6 to 5, and I said that's basically the same. So that's not conclusive. So then I need to ask the question, okay, what happens with the affected father? Here's an affected father. He has 4 children. Every single one of his daughters is affected. His son is not. Okay. Here's an affected mother. 2 out of 3 of her sons are affected, but 1 is not. Remember, that should be close to 50%. So given the fact that we're leaning towards females, that the affected father, all of the daughters and not the son, and then this pattern right here, this all suggests strongly it's X-linked dominant. It's obviously dominant because every affected person has an affected parent. Now, what about Y-linked disorders? Y-linked disorders are disorders that are found on the Y chromosome, and this is sort of controversial. If you look in genetics textbooks, the example that you'll find is hairy ears, which is a phenotype in which there's a lot of growth within the middle, not the middle ear, but the external opening of the ear. And you'll look in textbooks and you'll see this as an example. I tried to hunt this down several years ago because I was looking for more examples of Y-linked disorders. The problem is that most of the nucleic acid sequence of the Y chromosome does not code for genes. except for the genes related to the development of the male phenotype. And as I was going through this, I found some research, some more recent research, that suggests that hairy ears is not actually transmitted through a Y-linked mechanism. So I've put question mark here because I don't have a 100% reliable example of this transmission. I'm not saying it doesn't happen, but it's murky if it happens. Other than maleness, obviously maleness is Y-linked. Ignoring maleness, there may be disorders like hairy ears that are transmitted in a Y-linked way. If it exists, what would it look like? Well, we would expect only those with a Y chromosome to have it, so males would be affected. An affected father must pass it to every single son, and it will not skip generations. So this one's pretty easy to observe in a pedigree. This father gives it to all of the sons and not his daughter. This father gives it to his son and not his daughter. This father gives it to his 2 sons and not his daughter. This father gives it to his 3 sons and not his daughters. There are no affected females. It acts in a dominant fashion because every father must give it to all of their sons. So once again, this may not exist. It may be that we'll come back in 5 years and say, yes, hairy ears is transmitted this way. But if it does exist, this is the pattern that we should observe. So that's a review of basic ideas of pedigrees, how you use them, and the various patterns that you might expect to see.